Hello, everybody out there. I think we're alive now, so I'm going to introduce ourselves. But first of all, to welcome you all to this session, which is called Life in the Soil Under Pasture. My name's Fidelity Weston, and I'm chair of the Pasture-Fed Livestock Association. As an organisation, we are a group of around about 600, mainly farmers, but we've got academics, vets, butchers, restaurateurs, all members. And really our main aim is to rear our ruminants from grass only. And the result is that we're really interested in what's going in under the soil. So as a group of individuals, I think this talk tonight is going to be really interesting for us as an organisation. I personally am particularly interested in it. I farmed for many years, we've been farming for 35, 36 years. And I have to confess to have only started walking around the farm with a spade in my hand and digging holes for probably the last couple of years. So there you go, for 32 years. Sorry, Andy, I know it's very depressing, isn't it? But anyway, the whole thing about life under the soil has really grabbed our imaginations. And I think as farmers, we can all appreciate how incredibly important this is, and particularly going forward when we're trying to reduce our reliance on chemicals, understand more about how we get natural good growth out of the soils. At home, we have school groups coming to visit, and there are 3 facts that I like to tell them now when they come into the barn. The first is that they might think that there's loads of animals and livestock out in the farm, but actually 95% of life under our livestock farm is under the soil. The second is that we probably actually only understand about 5% of what's going on in the soil. And to that end, it's really great to have 2 such experts on this session tonight. And my third fact, which I try and inspire them with, is to say that soil science is actually a really new science. And so as young people, they should really think about going into this as a future career. Andy has just told me another fact, which school visits again, and that is that we know more about the moon than we do about the soil. So for the next hour, we've got the opportunity to hear from 2 experts. Each of them are going to give presentations for about 20 minutes, allowing for 15 minutes of questions at the end. And we may well decide, if there's loads of questions, that we'll break out into a Zoom call following that. So we're going to start off with Professor Andy Neal, who's a microbiologist at Rothamsted. His work looks at how what we put into the soil in the way of farmyard manure, presumably cows pooping straight onto the soil, is central to the soil structure, organic carbon, and how the soils work in terms of porosity and the hydrodynamics that go on. There'll be a short break then for any questions directly to Andy about his talk, but then we'll go straight on to Dr. Felicity Crotty, who is looking at bigger organisms, and at the moment is particularly looking at earthworms as the main emblem of soil health. And she's seeking to identify linkages between healthy soils and healthy foods, and is looking at how land use above the soil affect the food web within the soil. So it looks like we're in for a really interesting time, and I think I could do nothing more than ask Andy to start his talk right now. Thanks very much, Andy.
Thank you very much, and hello everyone. It's very strange saying hello everyone just to a blank screen, but there we are. I've been hanging around the Oxford conference today, and it's my first time, and I realize what a exciting and I suppose innovative conference this really is, and I hope that I can live up to some of the other talks and the excitement that I've garnered from some of the other talks earlier on today. So yes, I'm Andy Neal. I'm a soil microbiologist, and I said yes to talking in this session about life in the soil under pasture, but the more I thought about it, and actually the more I've delved around in soil, it's very hard to compartmentalize soils. And I really see them more as a continuum nowadays. So although, yes, this is a session about life in soil under pasture, and I will be talking about pasture, a lot of what I'm talking about is comparing pasture to arable systems and really looking at the whole continuum of soil, but really focusing on the system as that's the most important part of what I'm talking about, and how the biology in there works and drives the whole process. Now, my title really is, it's describing how organic carbon really drives this soil system, but something that we've developed, a sort of theory we've developed over the last few years, which I will come to explain and talk about, is soil as an extended composite phenotype, putting the biology right at the center of a good, healthy, functioning and efficient system, soil system. So the first thing I want to say is, although we have been studying soil for quite a long time, and scientific reductionism has typically led to, I suppose, science in general studying the physics of soil quite separately, and then the chemistry of soil quite separately, and the biology of soil quite separately. And it really hasn't got us very far. And so what I want to start out with is really suggesting to you that soil is really a process. It's a collection of, a hierarchy really, of interconnected processes that drive what we call an emergent phenomenon that we call soil. And so it's very, it's not a good way to think of soil as just the biology or just the physics or just the chemistry. We need to think this about a system and the processes that drive it. But the other problem we have with soil, and I've got some data here from long-term experiments at Rothamsted, is that it's a very slow process. So understanding it within the life frame of an individual research scientist like myself, or a farmer having to manage this process, can be very challenging, because as in the bottom left here, if you look at organic, this is organic carbon accumulating in soil that was originally treated as inorganically fertilized arable, and it was then abandoned and allowed to rewild. And you can see that it takes approximately 120 to 160 years for carbon to accumulate and reach some sort of stable endpoint. And you can imagine, if you're working that soil for 20 years here and there across that whole timeline, it's very difficult to see changes in that soil. So it's very difficult to understand and appreciate our individual effects upon soil within our individual lifetimes. And I think this is one of the problems where society finds itself today. We're now seeing, when we look at degraded soil, the consequences of centuries of management, and some would say mismanagement. But we're having to deal with them now, and we need to understand how to get this soil back into a good, healthy state. The other point I want to make on the bottom right there is that texture is really important. Texture is a really important context for understanding organic carbon in soil. Sandy loams are not very good at storing and sequestering carbon. Clay is really all important. And you can see here, these are our sandy and clay loams that we've used at Rothamsted, and this is about 60 years of measurements of organic carbon. And you'll notice that a good sandy loam treated as permanent grass never over that 60 years manages to hold as much organic carbon as arable treated, so regularly tilled and inorganically fertilized clay loams. So understanding these processes is very important from understanding the soil system. But as we move towards mechanisms to reward farmers for sequestering carbon in those soils, some of these processes have to be considered in the approaches that we take to make sure that people are rewarded for the work they're doing and not penalized because they're trying to work with soil that will never hold large amounts of organic carbon. And I don't hear that level of subtlety discussed when we're moving towards rewarding farmers for the good they do and looking after their soils. So I really want to talk about processes with biology at the heart, and processes at very different scales to what we're used to talk about. So I want to get us in the mood. I want to take us into a journey inside a soil aggregate to really understand where the processes are occurring that I'm going to talk about in this talk. So this soil aggregate is about 2 millimeters in size. So it's a very small soil aggregate. And these images were, first of all, captured using X-ray tomography of the pore network in this aggregate, and then using the the modern skills of computer gaming were given a sort of nice skin to make it look like an appropriate inside of a soil aggregate. Now, these pores that we're looking at are about 100 microns in size or smaller. And this is the scale that we need to think about when we understand how soil functions as an efficient system. Why? For a number of reasons. Firstly, microbes, and that's what I'm going to be talking about in my talk, are of the order of 2 microns or smaller. So this is the sort of habitat in which microbial activity takes place. And this is where we find a lot of the sequestered carbon down at these scales. And there's a reason for that. And that's because most of the sequestered carbon in soil has been rather like the water that we drink in London that's been through several people's kidneys before we drink it. The carbon that's sequestered in soil has typically been in and out of at least 1 microbe before it becomes sequestered in soil. So the scales at which microbes are functioning and influencing soil are down at these micron scale pores. But perhaps most importantly, because of the scale of these pores, they are the pores that are holding water back once soil drains. So the water holding capacity of soil is governed by these scales as well. So this very fine scale, don't worry, we haven't fallen out. We're going to go back in a minute. These are the scales that we need to think about when we think of soil functioning as a process. What I want to do in the rest of the talk is tell you how carbon is involved in this and why it's so important from the conceptualization of efficient soil systems for agricultural uses. If you want to see the end of the video, the link's there, but I'm going to move on. You can come back to it and see it, and when you do fall out the other end of the aggregate. So the basis for these experiments that I'm talking about are a long-term experiment of soil that has been terribly abused. It's had all organic carbon inputs removed for at least 50 years. So over 50 years, no plants have been grown in it, and to our best management capability, all plants have been removed over that 50-year period. And as you can imagine, it's in a very poor state, it's highly degraded, and it actually has very little carbon in it. What we've then done is planted and returned that to a grassland managed system or arable system and look at how carbon accumulates and the consequences for carbon accumulating in that soil. Now, what I want to show on the left-hand slide here, left-hand image here, if I can find my pointer, is that this degraded soil has only about 20 tons per hectare of soil in it. And using X-ray computed tomography, we can measure the connected porosity of the pores that we've just been looking at in the movie in that soil. And it's associated with very low connected porosity. But as we increase the amount of carbon that's gone through that soil, up until we get long-term arable systems that contain about 45 tons per hectare and have been managed that way for over 60 years, and then grassland, permanent grassland that's been in that state for over 170 years that contains about 80 tons per hectare, we see this gradual but non-linear relationship between the amount of organic carbon in soil and the connected porosity that is generated in that soil. Now, going further, we can get an idea for what this looks like. If you want to look at the two extremes, so this computed tomography image at the bottom here is taken from this very poorly degraded soil. At the top here is a computed tomography image taken from grassland soil. In a minute, you're going to see some yellow pore networks in these, and you'll see that there's a really stark difference in the amount of pores, and also the size of the pores and the connectivity between the pores. And as anyone who runs a logistics business or anyone who spends their time on social media will know, connectivity is really important for networks. And so you can see, as a consequence of these different managements, that very low carbon input soils have very poorly developed pore networks. They're very small pores and their connectivity is very limited. But in a permanent grass system, in a pasture system, the pore network is highly developed, highly connected, and it's a very different manifestation of soil. Now, why is this important? Well, the connectivity between pores drives a lot of the characteristics of soil. So here we've plotted the connected porosity of in brown here, the very highly abused soil, the sort of the bare fallow soil in yellow, in the mustardy yellow in typical arable soil that's been treated as continuous arable for 60 years, and then in green in continuous pasture that has been in that state for over 170 years. The data points are scaled to reflect the amount of carbon in there, and you can see that there's a strong relationship between the connected porosity and the hydraulic conductivity of those soils. Now, just to put this in context, as you increase connected porosity across the bottom here, you increase the soil's capacity to store water. This is your water holding capacity of soil, and it's intimately related to the volume of pore networks in the soil. As you increase your hydraulic conductivity, you're increasing the capacity for that soil to move water around. Typically, of course, to plant roots, but associated with that water is also a lot of nutrients, both for plants, but also the microbial processes that are going on in soil. Now, soil is also really cool in the sense that it functions like our lungs. It provides a very high surface area for water and oxygen to be in intimate contact. And so this hydraulic conductivity also allows oxygen to move through the system. And what I want to impress upon you in the rest of this talk is just how important this movement of oxygen through the system is for microbial metabolism and the efficiency of nutrient turnover in soils. So we have this nice sort of two-dimensional, in fact, three-dimensional, because we've got carbon in here as well, this three-dimensional visualization of how soil functions physically. And from that, we can model the amount of anoxic space that an individual soil holds. Oh, and here we've modeled it over a range of matrix potential from very dry soils on the right there at high matrix potential of about 40 kilopascals, all the way down to saturated, probably flooding soils at about 10 kilopascals. And what you'll notice is that the bare fallow, so that's the soil that has very little carbon inputs, a very poorly generated pore network, actually supports a very high volume of anoxic space. Anoxic meaning a lack of oxygen. So even at field capacity, which is about 30 kilopascals, approximately 1/3 of that pore volume in that soil is anaerobic, has no oxygen associated with it. And you see that the profiles for the bare fallow, the arable, and the grassland are starkly different. And the grassland maintains a very low total volume of anoxic space right up until the very saturated end of this figure. So the way that oxygen moves and the availability of oxygen through the system is drastically affected by the connectivity of pores and the volume of pores in soils. And this is all connected to the amount of amount of carbon that is put into soil and is turned over by microbial activity. Now, you don't have to walk away with an encyclopedic knowledge of this figure. It's more for my benefit than yours. But what I want to say is that we now have the capacity and the technology to be able to understand the abundance of microbial genes in soils to the extent that we can now understand how this three-dimensional pore network and the ability of it to control the movement of oxygen and nutrients through the system influences the abundance of genes. And what I want to pull out particularly here is that as you move progressively from grassland, which is a highly poor connected system, through arable to the very degraded bare fallow soil, you get an increase in the genes associated with anaerobic processes, which mirrors the increase in the volume of anaerobic space in those soils. Now, the reason we're interested in anaerobic processes is, first of all, anaerobic processes are very wasteful. They don't incorporate nutrients into biomass. It's actually a process that uses sulfur, nitrogen as alternative electron acceptors. The other problem with anaerobic respiration, other than its lack of efficiency, is that it drives off methane and nitrous oxide from soils. the two, one of the two, or one of the three, sorry, two of the three is what I'm trying to say. Two of the three important gases when we think about climate change and the greenhouse effect in the atmosphere. So not only are we getting a wasteful use of soil nutrients and effectively a loss from the system as nitrous oxide or methane and hydrogen sulfide come to that, but we're also exacerbating greenhouse gas behavior in the atmosphere. So anaerobic soils are actually in a very poor state when we consider nutrient use efficiency and the effects upon a wider environment. So all of this that I've just presented, all of this adds up to our view of soil as an extended composite phenotype of the microbial metagenome. Now, you may not be familiar with the term extended phenotype, but I would probably bet a lot of money on the fact that we're all actually implicitly understanding of what it is. Even the worst ornithologist out there can probably tell the difference between a blackbird's nest and an osprey's nest, or a ostrich's nest and a bowerbird's nest. In fact, they're so good at being able to tell them apart, the birds don't actually need to be in their nest. We can still tell the difference. This is a manifestation of a composite, of an extended phenotype, and that is the expression of the genes of an organism outside the boundaries of the organism. The nests that birds produce are actually driven by or coded by the genes that they possess. And they evolve. They have to evolve, because you can imagine a bird that builds a nest that allows eggs to fall out right, left, and center will not get to share its genes very effectively. So although the effect of the genes is expressed outside the body, they are still under control of natural selection. And this is what we're seeing within soil, not birds' nests, but something that we call a process-formed state, which is effectively, it looks a bit like a nest. It is the collection of pores in a soil that are built by the microbial turnover of organic inputs into that soil. And as those carbon products are turned, sequestered into soil, soil moves through this three-dimensional space that I was discussing a little earlier. Now, what's particularly interesting is that for a period of connected porosity, you can increase connected porosity quite a lot with a very little increase in hydraulic conductivity, until you get to a critical point, which we term a phase transition, where now with only very small increases in connected porosity, you get huge increases in hydraulic conductivity. I would argue that we need to tailor our carbon inputs to keep soil on the right-hand side of this phase transition, to keep them in highly connected, aerobic, efficient systems. And that's all driven by the amount of organic carbon we put in there. But this is actually a very positive thing, in the sense that the work I've just shown you, demonstrates that we can manipulate this process and we can engineer soils to be in that highly efficient, connected, well-oxygenated state, if we have sufficient organic carbon to hand and use it wisely. So this work was published last year. For those of you who want to look at it a little bit more, the reference is down there in the bottom left-hand corner. But you may be saying, well, yeah, this is all nice and academically challenging, but what are the practical implications of this? So,
Andy, can I stop you to ask just a few technical questions that come up on that bit of your talk? Just really quick ones.
That's fine. I'll try and answer them.
The first one is, are you measuring living and dead organic carbon? Each microbe is made of carbon, with 7 billion microbes in a healthy soil. That's a lot of carbon. Any cultivation surely sets microbial populations and soil carbon back.
Yeah, so if I go back to here, so the organic carbon that we're measuring here is actually measuring the total amount of carbon in the soil. So that does include microbial biomass, as well as microbial necromass, as we call it. So that's the dead part of the microbial community, plus any plant material that's put in there. So we're measuring the total amount of carbon in there. And as I say here, we think it's actually soil flux, the carbon flux, that's most important, not the standing stock of carbon. Is everyone gone? Can you hear me? Should I continue?
Yes, can you hear me now?
Yes, I can now.
Yes, we're back again.
Another question?
The other technical question is, read the graph of anoxic proportion versus water content. Is that proportion of total soil mass or just the pores?
That's the total of the pores.
Okay.
So we're modeling the amount of oxygen in the pore network under a metabolizing soil system. So accounting for microbial activity.
Okay, that is absolutely great, Andy. And I think there are a couple of questions here asking about whether we can make all this happen more quickly through regenerative farming practices. And I think that's what you're about to go on to, isn't it?
Well,
Is it?
possibly.
Well,
so I'm not going to talk about rates, but what I am going to talk about is how important farmyard manure, so cattle, the byproducts from cattle, is actually really important to nutrients systems. So we've got a similar analysis up here. So this is from the world's longest experiment at Rothamsted called Broadborg. Many people will be aware of it. It's a continuous arable, continuous winter wheat experiment that's run for over 170 years. So it provides us a really good insight into what long-term management or mismanagement of soil nutrients and soil carbon can do to soil. Now, I'm going to rush through this, but I want to demonstrate that if we look at the anoxic proportion in inorganically fertilized soils, and this is a soil that receives 144 kilograms of nitrogen per hectare, along with phosphorus and potassium, and compare that to soils that receive 35 tons per hectare of farmyard manure, you can see that their anoxic proportions in these soils are vastly different, especially at low water contents. Farmyard manure has to get up to about 60 or 70% saturation before it contains as much anoxic proportion as inorganically fertilized soil. Now, if we look at the genes that are associated with nitrogen metabolism in these soils, we see that, so these two soils are soils that were abandoned about 170 years ago and allowed to regrow and were turned into grassland and a woodland. And so we're now not managed agriculturally. And you can see those soils have a very different complement of nitrogen. I should say this heat map goes from blue, which is a very low abundance, to red, which is a very high abundance of individual genes. The genes are in columns and the treatments are running across as rows. So these unmanaged soils have a very high abundance of genes that are driving assimilatory processes. So that's turning nitrogen into amino acids and proteins, but more importantly, fixing that nitrogen within the soil. When we move to the arable systems, which are this group at the top, they are predominantly containing a lot of dissimilatory genes. These are the genes that are driving the anaerobic respiration and driving off nitrous oxide into the atmosphere. So we can see that our manipulation of these soils drastically changes the genes associated with nitrogen metabolism. Now, we were very interested in what is this causing, or what's the effect of this upon the loss of nitrous oxide from these soils. So we measured in 2019, we measured nitrous oxide emissions, not every single day, but over a long period between April and November, over a large number of days. And what we were able to show is that the farmyard manure, although it contains, or it receives about 240 kilograms of nitrogen per hectare per year as inorganic farmyard manure, loses considerably less, significantly less, than the inorganically fertilized soil that receives an equivalent amount of inorganic nitrogen. So although they're both receiving equivalent amounts of nitrogen, the farmyard manure loses much less to the atmosphere and retains much more in the soil, and now contains 3 times more nitrogen over the 170-year period. It's accumulated 3 times as much nitrogen in the soil than the inorganically fertilized farmyard manure. So that's looking at nitrous oxide losses over 2019. If we model the nitrogen balance in these soils over a 30-year period, you can see that the 3 inorganically fertilized soils here with either 144, 192, or 240 kilograms of nitrogen inputs have a very different distribution of what's harvested in the grain and the straw in green, what's accumulated in the soil in a reddy brown, and what's lost to the atmosphere in gray. You start increasing your nitrogen. Of course, you increase your amount of nitrogen in your harvest, but also you start losing more nitrogen to the atmosphere. That breakdown is completely different when you look at farmyard manure over the 30 years and see that actually less nitrogen is lost to the atmosphere and more nitrogen is accumulated in the soil systems. Of course, this is all driven by the fact that we have a much greater aerobic volume of that soil generated by the improved pore network in the system because of the enhanced carbon inputs. So carbon has a direct relationship to how nitrogen is managed in systems and how we can generate more nitrogen efficient processes for our agriculture. So just to summarize, we've got what we're calling the extended composite phenotype model of soil. We think it's a radically different view of soil as a critical system. That's one that exists somewhere between the boundary of chaos and monotonous order. So it's a complex critical system that accounts for organic carbon flux through the system, which allows microbes to generate microstructure, which has a strong implication for the hydrodynamics, and then controls microbial metabolism in turn in a big feedback loop. These systems can be manipulated to reduce the losses of greenhouse gases and increase resilience. The more water that's stored in this pore network, the more nitrogen that's stored in those agricultural soils on Broadwalk, the greater resilience the crops have to losses for drought or for limited nitrogen inputs. Grassland and grazing livestock have important roles to play in maintaining these systems. Grasslands actually contain the highest amount of carbon that we've looked at and the greatest and most extensively developed pore networks and the most assimilatory genes in these nitrogen assimilatory genes in these systems. But of course, the context is important. This is where we came in. Soil texture has a strong influence on this. Clay is generally good. Sand is bad, and we need to understand if we're going to have limited amounts of carbon we can use, we may not want to be investing it in sandy loams, and we should look for clay loams to invest it in where it's going to be sequestered most effectively. And we have to think about long time scales to do this. So ultimately, what I want to say is the judicious use of organic carbon sources is necessary, but we can manage soils for improved efficiency and performance, and it's all dependent upon this extended composite phenotype of soil as a biological living system. And I'm going to stop there.
That's absolutely great, Andy. Thank you very much indeed. There have been a number of questions coming in, but I'm going to keep them all now until we've heard Felicity, because I think they'll probably overlap with some questions to her as well. So over to you, Felicity.
Brilliant. Hopefully you can see my screen. Yes?
Yes, we can see your screen. That looks great.
Brilliant. Thank you. Okay. Hi, and thanks, Andy. That was really amazing talk and links really well with what I'm going to be talking about now. So when we discussed this session, talking about the micro and the macro, so I'm going to be concentrating more on meso and macrofauna within the soil. So hopefully you will find this interesting as well. So Andy mentioned it just at the end there, grassland soils have the highest carbon stock of any UK habitat. And part of this is to do with the fact that they are a highly stable environment. It's very much unlike in arable soil, the soil remains on the land and the plants stabilize it through their roots, returning nutrients, returning organic matter as they grow and die and grow and die. But the main organisms that are involved with this are the soil organisms that help through this process. Now, when you think about carbon, soil carbon, actually it is soil holds about 3 times more carbon than we have in the atmosphere. So we talk about greenhouse gases, we talk about the greenhouse effect, but actually if we released all the carbon within the soil, this would be a huge flux of carbon and highly polluting. So we need to try to keep the carbon within the soil as much as possible. Now, within the England, about half of the carbon is stored in just the top 30 centimeters of soil. So it's really, that is the key part that we need to look after. And so how resilient the soil is and how much we look after the soil will be affected by other variables. So for example, climate change. So over the last sort of 10 years, we've had a really bad run of extreme rainfall events, extreme temperature events as well. So 2012, for example, was the wettest summer for 100 years. 2013, coldest spring for 50 years. Then we had a run of warmest years on record as well. So 2014, 2015, 2016, 2017. Then in the UK, we also had Beast from the East, so large snowfall events where the polar vortex led to extreme cold temperatures. 2019 was warmer, wetter and sunnier than average. And we had flooding and record temperatures in Cambridge, and the Whalley Bridge nearly collapsed as well. And then in February 2020, I know that feels like a very long time ago now, there was storms Kira and Dennis, where we had again heavy rainfall and flooding and extreme problems across the country. Now, these extended periods of wet weather can cause widespread damage to soil structure. Heavy rainfall exacerbates soil erosion, leading to greater flood risk and polluting housing and also the surrounding area. If you're a farmer, if your soil leaves your field, then all of the nutrients, all of the soil carbon will also go. So you need to try to keep the soil on the fields, and that's why grassland is particularly useful, because it does help stabilize the soil. Now, what makes a healthy soil? So we quite often refer to soil health and we talk about soil quality, and there's Dorian Zeiss coined the phrase soil health or soil quality is the continued capacity of a soil to function. And usually if you're thinking about an agricultural context, then that would be the soil to function as for agriculture, for crop growth. But actually, when we're talking about soil health, we're actually, whether it's unconscious or not, thinking about soil life. You do not have a healthy table, for example. So if you're talking about the health of something, then you're obviously talking about the living system and not just an inert base for agriculture. And part of that living system is the soil organisms that reside there. Now, it's often been referred to as the poor man's tropical rainforest. This is due to the abundance and diversity of life within the soil. And the soil is home to about a quarter of all living species on Earth. That's identified species and unidentified. So huge amount of diversity. Now, these soil organisms are the ones that are driving the decomposition and nutrient cycling within the soil. And agricultural practice can change the soil habitat and influence the abundance and diversity of this soil fauna. So knowing what is there and how the soil fauna are working together will give us an understanding of the health of the soil. But there is insufficient data on this, on the health of our soils. And so investment is needed in monitoring this. Now, often in schools, soil science is often taught emphasizing the physics, the chemistry, without the soil biology part. But without the biology, it is just an inert substrate. The soil biology is one of the 5 main soil forming factors. So without life, it is not soil. So the soil on Mars, not soil. It's just parent material. So when we take into account this soil life, there is a huge amount in it. And Fidelity at the beginning of this session said that she said 95% of all animals, all life on her farm is below ground. I'd actually probably say it's even more than that, because when you think about biomass of soil fauna below ground, within 1 hectare of arable soil, you've got the equivalent of 5 tons of soil fauna, which equates to about 100 sheep. This breakdown is the equivalent of, say, a cow of bacteria, 5 sheep of protozoa, 24 rabbits of mesofauna. But what is mesofauna? Mesofauna are mainly springtails and mites. They're the organisms that are between 0.1 and 2 millimeters body width. So really, really, really small organisms. But these are the ones that are driving most of the nutrient cycling and decomposition processes. And just giving you a few examples of just the beauty and the amazing diversity you can see within the soil. These guys are springtails, they're microbivores, breaking down eating the bacteria and fungi on the organic matter, quite often making it smaller, making it available for, make it smaller, it's greater surface area, which means it's more available for the bacteria and fungi to process it as well. This guy, this is a Symphyleona springtail. This is the size of basically like a full stop on a Word document. So really hard to see in the wild. But if you do have some sort of macro photography, or if you've got a microscope, you can really get to see the detail of these organisms. Soil mites as well. We've got a predator on the left there and a decomposer on the right. Again, working within the soil food web, moving nutrients around the system. Final just picture show would be a pseudoscorpion here. You can see it's got a springtail within its pincers, just to give you an illustration of the size and diversity that occurs. It's always nice to have a few pretty pictures, I think, too. So these organisms are the main players within the soil food web. But when you think about biomass, there's actually the main player are the earthworms. And in fact, you can have a greater weight of earthworms alone below ground than you've got livestock grazing above ground, about 3 tons per hectare. And also, these guys are the ones that are improving your porosity within the soil, improving water infiltration. It's thought about 90% of all water infiltration wouldn't be possible without earthworms. So I was talking about arable numbers. In 1 hectare of grassland soil, you've got the equivalent of 50 tons of soil fauna, or 2,000 sheep. Normal stocking density is about 16 sheep per hectare. So huge, huge biomass below ground in comparison. Now, earthworms, these are often referred to as ecosystem engineers due to the huge effect they have on the whole system. The burrowing, the consuming of soil, the changing, cycling of nutrients and organic matter, moving it around the system, means that they change the habitat for the whole soil fauna as well as the soil itself. And it's sometimes it's been calculated that if you compared a soil agricultural land with earthworms and one without, you can improve your crop yields by 25% in the land which has earthworms. It's usually in a low nutrient environment. And actually knowing how many earthworms you have is a quick and easy gauge of the soil health. So if you took a block of soil, a spadeful of soil, and broke it up and counted the number of earthworms within it, if you had 16 per spadeful, that equates to about 400 per meter square, which is a large amount. And these ones, that would be the benchmark of a healthy soil and have these increased crop benefits. But we do need to think about what the soil type is and the time of year you're measuring. So a heavy clay soil would always have more earthworms in it than a sandy soil. Measuring in the middle of summer, when the ground's really dry, you're unlikely to find as many earthworms as you would in the middle of winter, or when the soil's quite waterlogged. So you need to think about those differences when you're monitoring the soils. Now, earthworms are split into 3 main functional groups. You've got your surface dwellers, the epigeic species. These live on the soil surface, really close to the soil surface. They're not really burrowing, but they're ones that are eating the recently fallen plant organic matter, recently fallen plant material, very small, dark worms. The second group are the soil dwellers. These are horizontal burrowers, burrowing horizontally through the soil profile. And these ones just eat soil. They eat the soil, they process the soil, and they make it a better soil because of it. Finally, the third group, you've got vertical burrows. These ones are deep burrowing anesic earthworms. They live a long time and create these permanent vertical burrows. And what they do is they take plant material from the surface and they pull it down into their burrow. And once it's in the burrow, then it decomposes and then they eat it once it's started to decompose. So they're responsible for moving the carbon and organic matter around the soil profile. So in the UK, we have about 30 species. In Europe, you've got about 300 species. And the reason for this is that when there was the Ice Age glaciation, when we were under ice, killed all the earthworms. And so when that melted and before the channel appeared or was filled with water, was when the earthworms crossed the channel and came to the UK. And as I like to say, I don't know if this is going to work. How fast are earthworms? So if you think about them crossing the channel, we're basically getting the fast moving species, the ones that are most active. And what you can see in this little silly video here is that actually we have got a number of different functional groups. We've got the paler earthworms there, the horizontal burrowers. We've got the small black worm there in the center. That one is one of the surface dwellers, and the really large one, that's your vertical burrower. And what you can also see is that the bigger they are, it isn't the faster they move. But because of how healthy a soil is, will depend on having actually a mixture of all of these things, because they've got different food sources and different life history patterns. So where they will be, so the surface dwellers, quite a rapid reproduction system, because they live on the surface, very much affected by ploughing. The vertical burrows as well, also because they create these permanent burrows, likely to be most affected when, particularly inversion ploughing, cutting through the burrows, cutting through the worms themselves. Horizontal burrows, not so affected. So thinking about the distribution of earthworms, as I say, so this is a paper that I worked on a couple of years ago now. You can see that actually earthworms also show an opposite to most organisms, or soil organisms in general, actually. And that's you actually have greater abundance and diversity in temperate regions, so UK, Europe, in comparison to the equator, in comparison to rainforests, which is the opposite. And it's likely to do with the amount of soil carbon as well, because there's more soil carbon found in temperate regions. So there's this linkage here between the two groups. So briefly, just to finish off, I'm just going to talk about a few experiments which have looked at how agricultural management has affected soil organisms, but particularly earthworms. So first of all, looking at different forages. So we worked on investigating the impact of different forages on soil biodiversity, and whether they're with replicated plots. You can, and if you grow these forages up over time, you can start to assess whether there has been a movement of invertebrates from one plot to another. So for example, with this one, so there was ryegrass, chicory, white clover and red clover, replicated blocks. These were growing for 3 years before we looked at soil fauna. And what you can see from these graphs, the graph on the right, graph on the left, sorry, looks at total earthworm abundance. And in this one, we're just counting total number of earthworms. You can see there's a huge number in the white clover forage treatment in comparison to the others. So you're getting about 800 per meter square. So huge amount, a huge number of earthworms. When we broke that down into looking at the different functional groups didn't really show a pattern. But when we looked at the different species, you can see that there were 3 main players, the graph on the right. And so that's Caliginosa, which is a horizontal burrower, vastly higher numbers in the white clover treatment, as well as Lumbricus terrestris and Aparatosia longa. So both of those being vertical burrowers. So again, main increases found in the white clover treatment. Now, what happens when you start to increase your agricultural management, when you start to increase your intensivity? What ends up happening is you're selecting for organisms with smaller body size and lower biomass. So you'll always find greater abundance and diversity in an extensively managed system than an intensively managed system. So what I want to just describe briefly next is a few long-term experiments that have been set up at RAU. So this looks at 3 treatments, plough, minimum tillage and direct drill, that have happened on replicated plots every year for the last 10 years, basically. And initially, the plough-based treatment had the highest yields and direct drill was lowest. But what then started to happen was that we saw a significant correlation dependent on the weather conditions. So in low rainfall growing seasons, direct drill was the best. High rainfall, it was plough. But soil biology had not been continuously monitored, although now actually we've got a master's student and who is now starting to really delve into soil biology. But what I thought I would do is do a quick discussion of the earthworms that are there. So looking at the overall abundance of the earthworms is quite similar between the plots, but what we found was that there was this a selection pressure for there being, so there was much less surface dwellers and vertical burrowers, the anesics, so epigeics and anesics were much lower numbers in the plough treatment in comparison to direct drill and minimum tillage. There was no difference between those. Also that the numbers were quite low in all treatments, and this is likely to do with the drought. So we've had a number of spring-summer droughts over the last 2 years, 2018 particularly bad, 2019 as well was quite bad. When the soil is really dry, earthworms aren't able to feed. So they're basically, they end up either burrowing really deep or they create these little sort of cocoons within the soil, and so they keep themselves, to keep themselves wet because they breathe through their skin. So spend a lot of time not eating is likely to end up dying. So this is why numbers have been reduced lately. And as I say, it's just a hugely important number of functions that they create, have within the soil. Now, just to move on slightly, and something that's a little bit of a bugbear of mine is that a lot of people talk about no-till versus tillage. And actually, not all tills are the same. And so at RAU, we had a 5-year demonstration experiment where we had in each field a different establishment method. So we had a plough, and then we had 3 direct drills. So we had Missouri, Seedhawk, and cross slot. And what you find is actually there is again this change in number of earthworms. You can see that although overall abundance, no difference between treatments, again the plough much reduced for the epigeic species and also the anesics, the vertical burrowers, with the Missouri and cross slot being much better and the Seedhawk intermediate. So what you're seeing is that actually all the direct drills aren't all the same. However, there were differences in soil structure as well, which might link up with this soil moisture effect as well. So if you think about this was in quite a dry year. And again, with the soil structure, you can see that there's real differences between the different drills. So I realise I'm quite short on time now. So I'll just to finish off. So earthworm abundance has been shown to vary due to crop, so the forage species linked to food availability potentially or food preference, and also crop establishment methods. And so if earthworms are being used as an indicator of soil health, then we need to consider these external drivers before we really, make the point that it's all about the earthworms. And just to reiterate the point that the healthier soil is, the more resilient it will be to these future weather extremes. So thank you. I think there's some references there which you can look at in your own time, and hopefully there's something for you to think about. Brilliant.
Thank you very much, Felicity. That was fascinating. I loved the worm race. I mean, 2 speakers, one with a video of soil and another worm race. What an amazing speakers. we decided that we would have a breakout session, perhaps to discuss some of the wider issues over what we as farmers could be doing on top of the soil to affect what goes underneath the soil. That would be an interesting discussion to have with Felicity and Andy. So if people want to join us for that, we have arranged for that to happen. So I think in the meantime, what I'll do is just ask these questions directly for you, Felicity, that have popped up on the questions. So here's the first one, which is that from somebody who says that they always find far more earthworms in digging up undug pasture than very well manured and composted organic veg beds. Is this because the soil is healthier under grass, or because worms dislike cultivated vegetable beds and burrow deeper?
I think it might be a combination of factors. So if they've been very well cultivated, then of course you're digging up the system. So you're going to be disturbing the house that they live in. So you're going to be mixing the soil. What's likely to happen is when you that they might move back in afterwards to utilize the compost. The other thing is that it could be that you're affecting the acidity of the bed. So if you've put in a large pulse of manure, for example, slurry is particularly bad, although that wasn't part of the question, but it will release a lot of ammonia, which can reduce earthworm numbers in the short term, just because of the pulse of ammonia. So it's something to think about. But the pasture, you will find a lot because of the stable environment and because of the continuous accumulation of organic matter. You're giving them the food and you're giving them the habitat. So you're always going to find large numbers of earthworms there.
So would you say, Felicity, that there's greater soil functionality and species diversity on undisturbed long-term grazed areas such as SSSIs and high nature value land? Those soils, that there is a strong correlation that they haven't ever been ploughed or disturbed. So that is one question. I'm going to ask you to also pick up on the impact of power harrowing on surface earthworm populations. And also another question here, which is that if we have only got 10% of the species of earthworms in Europe, is there a case for introducing more species to the UK?
Too many questions. I think the first of all, the introducing species is usually a bad idea, would be what I will just say. I'll just put it out there now. So when you think the UK has 10% of the species from Europe, USA has 0%. So USA has a large problem with earthworms that have been introduced. So Lumbricus terrestris, the gardener's friend, this huge vertical burrow that does all this good in the UK and in Europe, terrible in the USA. It's destroying forests because it's so good at processing all of the fallen plant material. So the environment has grown to be used to the earthworms that are there already. So introducing them would be a bad idea, in my opinion. As to cultivating with a power harrow, similar to I specifically haven't looked at power harrowing and reducing earthworm numbers. But when you think about what earthworms need and what's the main problems with ploughing, for example, which power harrowing is similar, is are you going to be physically damaging the earthworms themselves? Potentially, if they're near the surface. Are you turning their world upside down? Are you cutting up the burrows that they make? So surface dwelling earthworms, you wouldn't be cutting up the burrows because they don't really make any, but you might be damaging them physically. If you're changing their world, moving their habitat around, what happens? You're then going to be making them much more open to the atmosphere, much more liable for predation, and also potentially changes extremes in temperature. So if it's a very hot day, they could dry out and basically turn into earthworm crisps. If the water table is really high when you're doing it, then they're more likely to be near the surface as well. So you're more likely to be damaging a lot more of the earthworm population.
Thank you, Felicity. On that note, we're going to have to very quickly end. We've got a minute to go. So I'd like to thank you both very much indeed, and everybody who's listening. And just to say that, please, if you want to join in the discussion on some of the other bigger questions about what we're doing on the top of the soil, I think that our wonderful volunteer who's been facilitating the questions will let us know what to do to move over to the breakout room. So look forward to seeing you there. Thanks, everybody.